prenatal diagnosis program, and is also the Setting Chief of Gynecology. He's also Assistant Professor of Developmental Medicine and the Graduate Institute of Clinical Medicine of Tzu-Chi University. Today, his topic will be human umbilical cord mesenchymal stem cell support prolonged expansion of human embryonic stem cells without tumorigenesis. Let's welcome Dr. Ding.
Thank you, Chairman, for your kind introduction. First, I would like to thank the organized committee to invite me to present our recent work. My topic is human umbilical cord mesenchymal stem cell can support prolonged expansion of human embryonic stem cell and without tumorigenesis. So the origin of stem cell can come from embryo, fetus, and adult. So the stem cell, yesterday many speakers said that the definition of stem cell can be a semi-renewal and can do a differentiation. So different stage of stem cell can have a different potential to differentiation. So for example, if your stem cell derived from the embryos, such as inner cell mass of 5 to 7 days embryo, the stem cell will be totipotent. It can differentiate to any kind of stem cell in your body. If we obtain the stem cell from the fetus, also prepotent or multipotent. If we got the stem cell from adult, from skin or neuron, it will be multipotent. So these different kind of stem cells, like embryonic germ cell, embryonic carcinoma cell, they will be prepotent stem cell. So different stages have different differentiation potential. The ES cell, embryonic stem cell, have prepotency. It can differentiate to any kind of cells in the body. So ES cell derived from the blastocyst, the inner cell mass. Inner cell mass will differentiate to 3 germ layers, cells, and germ cells. So the ES cells, the prepotency is very important for future regeneration therapy. You can differentiate any cell you want. So then IPS was found in 2006 by Yamanaka. So he added 4 factors into the fibroblast and can make fibroblast into the IPS. So this is a new kind of stem cell and look like embryonic stem cell, but the gene expression is a little different from the embryonic stem cells. So there are many kinds of stem cells. So the future, the promise of the stem cell research, we can use it for identifying drug targets and test potential therapeutics and toxic testing. Like many drugs, if you want to use the fetus, so the toxicity testing, or you want to use it for regeneration therapy, you can differentiate to many kinds of stem cell. Bone marrow stem cell, nerve cell, heart cell, or pancreatic cell. This regenerative disease is now we don't have a good method to cure this kind of regenerative disease. So the stem cell give us a future that if we can processing the embryonic stem cell very carefully and differentiate to specific cells, you can use the useful stem cell therapy in the future. And also we can use the stem cell to study the cell differentiation, understanding prevention and treatment of birth defect. You can use for a lot of aspects. Provide the ideal research method in developmental biology and cell therapy. So it is if we can maintain the ES cell or derived stem cell or derived the iPS cells for this kind of various aspect, we can use for many, can do a lot of research and maybe use for therapy in the future. Originally, the embryonic stem cell was cultured on inactivated mouse embryonic fibroblast, MEF. So previously paper reported That ES, if you use ES cultured on the MEF feeder layers, you may contaminate animal proteins, such as new GC is acetic acid. So human cannot form new GC, new 5GC protein. So if we use this kind of stem cell to implant, transplant to our human body, it will cause the rejection, immune rejection. So this kind of problem should be resolved before the stem cell transplantation. So later on, many different human feeders was first is the foreskin cells and endometriums and the fallopian tubes, the cell, and the bone marrow stem cell, human-derived fibroblasts, and the fetal muscle skin cell, placenta amnion. They are reported these feeders can maintain the ESL prepotence. But if you use this kind of cell, you must do surgery before you get the feeder cells. So, so later we will introduce our the feeder cell origin is from the umbilical cord. It's non-harm. You can you don't have to do the surgery to get these cells. And how do you characterize human embryonic stem cells? First, you can see the morphology is colony formation, colony growth on the feeder cells, and the passage, many passages, the chromosome stability is maintained. And we test, and you must test the prepotency gene expression. This is alkaline phosphate, OCT4, and SES3 and 4 expression. Then you do the embryo body transformation and to do the differentiation, induction in differentiation. So you can in vitro differentiate into 3 germ layers. And later, the later step, you must put into the non-skin mice and form the teratoma and form 3 germ layer cells. So do all these things, you can claim that your cells is human embryonic stem cells. So the purpose of this study, if you want to use the human embryonic stem cells in human therapy, animal-free and tumorigen-free culture system is needed. So we want to find a new human feeder cells to eliminate the animal protein contamination. And the second, want to diminish the chance of teratoma formation after transplantation. There are a lot of origin. People will say a lot of origin of stem cell, bone marrow stem cell, neural stem cell, OEC stem cell, skin stem cell, blood stem cell. A lot of stem cell. So previously report they find stem cell from Wharton jellies. Wharton jelly is umbilical cord stroma. You can see this Wharton jelly site. This umbilical vessel, the adventitia, and Wharton jelly and the amnion. So this site is rich of stem cell. They can novel source of stem cells and can differentiate into neural-like cells, both in vitro and in vivo. So I'm a gynecologist, so we have a lot of source of the umbilical cord. So we want to try to derive the stem cells from the Wharton jelly. So in previous report, we derived stem cells from the Wharton jelly and put into the rat stroke model, and it can improve the stroke rate behavior. So because we have a lot of stem cells from the umbilical cord, so we want to use to test if it can to be a feeder for the human embryonic stem cell. So this is a big picture. This is Wharton jelly, adventitia, umbilical vessel. So we want purely stem cell from this Wharton jelly site. So first we collect cord. This is the placenta and the umbilical cord of human fetus. This is a cord, and we cut in pieces and remove the vessel and derive the stem cell from the Wharton jelly. And we cut Wharton jelly into pieces. Then the cell will grow. After 7 to 14 days, it will grow out from the pieces. So this is a histology of umbilical cord. We can see different CD marker expression. And this is a stem cell spreading from the cutting pieces of Wharton jelly. And we also test the human umbilical stem cell for cytometry. CD34 negative, CD90 negative, CD34 negative, and CD45 negative, and CD44 and CD90 positive. This is same with the bone marrow stem cell character. And when we do differentiation study of this kind of mesenchymal stem cell, you can differentiate neuron, adipocyte, and osteocyte very easily. So we can this mesenchymal stem cell from the human umbilical cord stroma. Then we use as a human umbilical stem cell feeder. You can see the growth picture is different from the ESL culture on the MEF feeder layer. The growth picture of ES MEF feeder, you can see it's really round in shape and good looking, healthy. But in Wharton jelly feeder layer, it's some different. It will cause like this shape, and the surround area could be differentiated cells. But this central side, the cells will be maintained its prepotency. Later we will see. So this is a magnification of this picture around this area. You can see high NC ratio. Nuclear is prominent in the cells. Maintain its growth picture of the embryonic stem cells. Then we compare the proliferation rate of the ESL culture on both feeders. You can see they are all the same, not much different between these two kind of feeder cells. And then we test if this ESL culture on Wharton jelly can maintain its prepotency gene expression. So we test this is alkaline phosphate, OCT4, SOS2 NALOG, SSEA4. We can see all the prepotent gene expression and maintain normal chromosome, female chromosome karyotyping after 20 passages on these feeders. So it's a good feeder, can maintain the human embryonic stem cells prepotency. Then we see if it can differentiate well, and you can form the embryonic body. Very beautiful picture of this. You can form a many embryonic body. This is the first step of differentiation. Then we put on the culture dish, and he will differentiate to a 3 germ layer cell. This is a neurofilament 200 MAP2. This is ectoderm neural stem cell, and this brachyury is mesoderm, and the ATBF1 is endoderm differentiation. So 3 germ layer differentiation also can conduct in this kind of human embryonic stem cell culture on Wharton jelly. You can do the cardiomyogenic differentiation. We look RT-PCR of this gene expression in ES culture on different feeder. You can see, however, this ARC4 gene is is less expression than the culture on MEF. And germ cell, endoderm, mesoderm, ectoderm differentiation also noted in the EB, different from different cultural conditions. So late, the last stage, you claim this feeder is a good feeder. You must do the in vivo teratoma generation. You can see this table. You can see after we get the ESL, we test on the we cultured on, because originally this kind of embryonic stem cell is cultured on the MEF feeder layers. Then we test the teratoma injection. 100% can generate teratoma. After we removed to the this kind of human feeders, mesenchymal stem cell from the Wharton jelly, you can see we do more than 1 year experiment, cannot get any teratoma formation. So we felt very frustrated. why he cannot formation teratoma formation at this feeder. So maybe this feeder is not so good, but after we transfer to the MEF feeder, you can see you also can generate teratoma. So culture on this kind of feeder can maintain its prepotency, but it cannot form teratoma formation. So this gives us a chance to test why this human embryonic stem cell cannot generate teratoma in vivo. This is how we do the teratoma formation experiment. Different dosage and different kind of mice and different different injection site. We cannot get teratoma formation when the human embryonic stem cell culture on this kind of feeders. This we transfer back to the mouse embryonic fibroblast. It can form the teratoma, can form the 3-germ layer teratoma histology. So later we suspect, speculate if there are more differentiated in our feeder. So we use the ARC4 GLP ARC4 drive GLP promoter, and we use lentivirus to transfect the human embryonic stem cell to get OCT4 expression ES cells. Then we inject into the non-c-myc, still cannot get teratoma formation. So we try to figure out what the cause this kind of human feeder cannot cause the human embryonic stem cell to generate teratoma. So we look at one c-myc gene. This gene is one of the IPS gene. So we look the c-myc gene expression on this kind of ES cell cultured on the Wharton jelly cells. So we can, the c-myc gene is less than half of expression cultured on the MEF. Then we do another experiment to use the Wharton jelly cells co-injected with cancer cell line. It will cause tumor formation less than the injection pure cancer cells. So this is a volume of tumor volume when you inject these cancer cells. This volume is smaller than the pure cancer cells if you add Wharton jelly stem cells. So Wharton jelly stem cells seems to have ability to cause the tumor formation will decrease. So this feeder, mesenchymal stem cell from the human umbilical cord, this feeder can support continuous sustained culture these ES cells for more than 20 passages. So now it's more than 100 passages. This gives a significant reduction of workload because you can, if you want to maintain the human embryonic stem cell, you must get the feeder often. So if you often want to derive the feeder cells, they will increase the workloading in the laboratory. So this kind of stem cell is easy to get and easy to derive and easy to maintain. So this will give to significant reduction of the workload in the lab. And also this kind of feeder can support continuous sustained culture of human embryonic stem cell more than 20 passages. So it's a good feeder. Then we wonder how this kind of stem cell can inhibit the tumor growth. So to look into the embryo development, you can see it's an umbilical from this side. It's embryonic tissue stem cells. So maybe correlate with the embryo development. He will not let the embryo to become a tumor in the mother's womb. So this is our speculation. We didn't prove yet. So this is our next step to study. And teratoma formation is a drawback of clinical application. Now, why the embryonic stem cell IPS cannot use for human therapy? Because it will cause teratoma formation. So it will block us to use this kind of stem cell for the human therapy. So how to decrease teratoma formation is our work. So the risk of teratoma formation due to contamination is small proportion of undifferentiated cells. So no matter what method we use, maybe flow cytometry to sorting the pure differentiated cells. However, due to technical difficulties, so there may be a small proportion of undifferentiated cells will contaminate in your transplant cells. So there are many methods to eliminate teratoma formation, such as transplant more differentiated cells. Like yesterday, Dr. Guo, he late stage differentiated progenitor cells. He may eliminate teratoma formation. And the surface marker sorting, you use Fox sorter to exclude the SCCA4 on different cells. However, the techniques maybe cannot achieve the 100% exclude. So this is a problem. And in our previous research, we found the DHA can eliminate teratoma formation in IPS transplantation. This part I did not show. So there are a lot of study to do teratoma formation. What causes the teratoma formation? In mouse embryonic stem cell, ERAS responsible for tumorigenic growth. This is well established. And recently, I attended a meeting of PPSC, SSC in Taiwan last Sunday. They report GLI gene is uncle gene, the downstream target of the Sony hedgehog. They find this gene may be responsible for mouse embryonic stem cell teratoma formation. In primates, embryonic stem cells, ERAS also expressed, but they in that study, they're not related to tumorigenesis in these primates embryonic stem cells. How about in human ES cells? ERAS is not expressed in human, so now it's not well known how the teratoma formation of the human embryonic stem cells. Then we do another study to see the c-myc in embryonic stem cells. We can see in our feeders, the c-myc expression is half than the MEF, than the mouse feeders. So c-myc is one of the IPS gene cocktail and one of the first proto-uncle genes, a potent role in most human cancers when expression is dysregulated. So it's a proto-uncle gene. So if it is dysregulated, it will cause tumor formation in our body. So the expression of c-myc is too high will cause cancer. Too low, you will yield non-functional stem cells. So the key to safely utilize stem cell in regenerative medicine may reside in maintaining appropriate expression levels, neither too high or too low of c-myc. This is a review of in cell stem cells. So in our study, the expression level of 100 cultured ES cells is between MEF and the embryonic body. It's a suitable level. We didn't know. We will do a lot of research study for this to resolve this kind of question. So our conclusion is this new feeder is a human origin. In previous study, he has a low immunogenicity and do not form teratoma in vivo, and therefore provides a significant advancement toward the future application in cell therapy. So I would like to thank our member in our lab, and Professor Drew, and our hospital, and our department, and the grant from the National Science Council. Thank you very much.